So my name is Tatyana. I'm a senior research fellow in Citron, University of Sheffield. I'm the group leader here. So my group focuses on on the molecular aspects of neurodegeneration and ALS in particular. So this project aims to develop novel small molecules, novel compounds that bind and target a novel molecular target that we've been developing for many years. It is called NEAT1. It is ribonucleic acid or RNA, and in cells it serves as a building block for structures called paraspeckles inside the cell nucleus, which are present in neurons as well. In my early studies, I found that the structures and this molecule, NEAT1, is accumulated in ALS, in motor neurons, in the surviving motor neurons. And we also found that this molecule is protective under stress. So our hypothesis was that paraspeckles and NEAT1 protect motor neurons, and they can be targeted or boosted to deliver a translational effect or to improve neuronal resilience and survival. So within this project, we want to develop the molecules we already have with from previous projects and previous screens. We want to develop them into better, improved or optimized molecules that can boost NEAT1 and achieve neuroprotection, and hopefully develop into a novel therapy for different ALS subtypes. This funding from the foundation is vital to further develop the molecules, because there is a clear gap in translational funding of this kind, and it's really important that we manage to leverage this funding, this project to take it further. So paraspeckle is a very small granule in the cell nucleus. We also call it condensate, and NEAT1 is really the scaffolding molecule for the structure. So without NEAT1, paraspeckles do not form. And paraspeckles levels go up under stress. That's why researchers believe that they might be contributing to protective responses under stress. And indeed, more recently, we show that if we get rid of paraspeckles, then neurons become compromised. So we now have sort of proof, research proof that paraspeckles are protective under stress. NEAT1 and paraspeckles a universal molecular target. So what I mean by that is we know that there are links, there is paraspeckle assembly and links between paraspeckles and the ALS pathogenesis across different disease subtypes, including sporadic disease. And we all know how challenging it is to develop new therapeutics for sporadic disease. So by developing these molecules, we hope to develop drug candidates that will be applicable across different disease subtypes. So it's really important in MND. It's really important to focus on such universal neuroprotective therapies that will be beneficial for a large population of patients. With this award, with this funding, we hope to take our molecules to the stage when they become really promising preclinical candidates and can be taken into further development. It's a lot of work, so it requires chemistry input, obviously, and we have chemistry partners on board who are also investigators on this award. So we have all the tools and expertise in place to deliver this. We hope that the candidates that we get at the end of the project will be drug-like candidates with really good potential for further onward therapeutic development. The short-term impact to generate two molecules that will drive the field forward and help the researchers to better understand the mechanisms of motor neuron disease. So we are committed to share our tools with the wider research community. So in the medium term, as I already mentioned, we want to deliver good preclinical candidates, which can be taken into in vivo studies. And in the long term, of course, we want to deliver orally bioavailable drug for ALS patients. And I think it is important that we are developing small molecules which have this really massive advantage that they can be taken orally, which is minimally invasive. Also make sure that we have high confidence in the target, and that's why we have, all those go, no-go decision points in the project incorporated, because we really need to revisit our findings and make sure that we develop only the promising molecules. This kind of funds are really vital for translational project like this one, because we have a lot of funding for mechanistic research, and then we have funding for development of clinical candidates. So I think this gap is covered by the foundation, which is really great.